Good morning, viewers. Welcome to today's lesson. And we remember last time we were talking about diffusion and facilitated diffusion. Those two were described as passive transport. And we saw that in those particular cases where we are talking about the passive transport, molecules were moving along a concentration gradient, which means that they must be concentrated more somewhere and then less inside the cell, and therefore they have to come in through the diffusion process. Now we agree that when these particular molecules were coming in, we said that the concentration gradient must be maintained, and therefore any molecule that has diffused across a membrane, assuming that this is the region of high concentration, and then they are getting inside a cell that is passing surface membrane, what could be seen happening is that the moment the molecules get in, there is that act of mopping. That means that this molecule has to be used up by the cell through the process that we can call respiration, for example, or a chemical process that use it up to make sure that the gradient is maintained. Or they can be seen being taken into the organelles, assuming that this is organelle. So this one will be leaving the molecules to be continuously less inside the cell, and therefore the concentration gradient, is retained because this is what this process was relying on for it to happen. So this is what we saw last time, how the molecule were being moved across a membrane. So that was the key important thing for us. So we really remember that in the process of diffusion or facilitated diffusion, there has to be a concentration gradient. This concentration gradient must be maintained at steeper level. So it must be steep gradient. And that one has been seen by any time the molecules get into the cell, it is being mopped up. What we are talking about, like taking it just like the normal mopping of the floor surface. You are wiping something out of the floor. So you are taking the molecules that has come into the cell, that is in the cytoplasm, and then you either take them to the organelles where they'll be used up, or we just use them in that particular area so that their number reduces, and that one was maintaining that was maintaining the diffusion or the facilitated diffusion, as we saw in the last chapter. Now today I want us now to look at it, that is this the only process that was taking place into the cell? We realize that this is not the only thing that was taking place inside the cell. There are some other molecules that must be taken into the cell, and this molecule may require the use of the energy, and that one takes us to the discussion of today, that we are talking about the active transport. So we can say that active transport is the movement of molecule. We can say that active transport is the movement of molecules or ions from their region of low concentration, concentration to their region of high concentration, That is the thing, that is passing across a semi-permeable membrane. That is the thing. So looking at it keenly, we realize that in this process, the molecules are coming from where they are less. In other words, they are going against a concentration gradient. That's another thing that's very important for us to note, that molecules in this process, molecules move against a concentration gradient. That is a hint that we must be keen with when we are addressing this particular subtopic, that there is movement against a concentration gradient. Now, when it happens that molecules are moving against a concentration gradient, it will therefore call for the use of energy. So therefore, there is use of energy. So this is the key thing, that molecules, for them to move against concentration gradient, we are going to use energy, and this energy is generated within the cell. So we talk about the cellular respiration has to take place here, so that we can get that energy. And in most cases, cells store energy. So we know that cells store energy. Cells store energy in a molecule called adenosine triphosphate. So this is a molecule that is required for the active transport to take place. And in this case, these particular molecules that are going to pass across the membrane, we also need what is called protein carriers. Protein carriers, which will be actually carrying these molecules across the surface membrane that we have talked about here. The molecules have to pass through the surface membrane, and then for that to happen, we need the protein carriers, which are always specific, because they have got shapes that are also specific. So these carriers here are specific to the molecule they carry. The molecule they carry. For example, if any case I have got a sodium ion here, the protein carrier that will be ferrying this across this membrane must be a specific one. Therefore, this molecule has to bind on the surface of the carrier so that it can be passed across the membrane. That is one thing that must be there, that is very much important for us to note. Now, number two, I want us to realize that when these particular molecules are being carried across the membrane, when they are passing, this is a store of energy, meaning that this particular store of energy, it has to release that energy. For it to release the energy, it has to be broken down so that we get energy out of it. So during the active transport, we also need, on the surface, we need to have a molecule that can actually break the adenosine triphosphate, so that it releases the energy to help the protein carriers to move. So what needs the energy is the protein carrier so that they can move. The source of energy is adenosine triphosphate, and then it cannot just work the way it is. It has to be broken down so that it can release the energy. Now, I have just said that for this to happen, we need that on the surface of the cell there is enzyme. Now, this enzyme is the one to facilitate the breakdown of ATP so that it can give us energy. So it will be breaking it down into two molecules, and then energy will be given out. So since we have learned in the earlier level of learning, we know that the enzymes, they get their name from the molecules that they work on. So if the molecule is ATP, then the enzyme will be ATPase. Remember, we were adding the suffix ASE, adenosine tripase. The ASE and ending of that one gives us the enzyme. So the work of this enzyme will be to break down. It is going to break down for us the molecule of adenosine, that is ATP. It is going to be broken down. If we have got the enzyme ATPase here, we are going to see that at the end of the day we have got an ADP, adenosine diphosphate, and then we also have got the phosphate molecule here coming out. So we have got the two things coming out, and energy will be given so that this energy will be used by protein carriers, and then it is going to carry the molecules across. Now this one, as can be seen, or we can illustrate the act of transporting this molecule across a cell membrane by drawing a diagram that will be showing us how molecules will be moving across the membrane. So I can illustrate that using a diagram. So assuming that I have got a surface membrane, up to now I hope we know the nature of the surface membrane. Like I have got this is my membrane, this is my membrane here. I'm just showing a simple sketch here to give us an illustration of how the molecule will be happening. So assuming that I have got the protein carrier here, assuming that this is my protein carrier, and then this is the side of the carrier, this is the shape that I was talking about. It has to be specific. I can have, this is another arm for it, and then I have the shape here. So that is the shape that will be accommodating the molecule that I am talking about. So this is how it is. So this is what I'm referring to as the protein carrier, and therefore I have got the molecules, assuming that they are glucose, for example, they'll be fitting in here. So we can have one being picked up there. So the molecules fit itself this way, and we are referring to as protein carrier. It is in the membrane. Now this is what happens. So these are the molecules to be actively transported across this entire membrane here, so that it comes here. So what will happen is that there is the shape here. The shape is definite shape. Then the molecules here, molecules will get into the shape here, and then that shape lock it up. Then how does it move? What will happen? Now this particular protein carrier, from the energy that it has gotten from the adenosine triphosphate after the breakdown, it will start moving. So it will turn. It has got the ability to turn, and then after turning on the surface there, it moves with the molecule, so that the molecule can be seen at the end of the day. The next step that we are going to see happening is we have the membrane now turning up, so that the molecules we are transporting, we see them changing the direction. So we have got this is now how the process works. This is now where we have the step 2. So up to here, what do we see? We are seeing that it has turned, so it now have this particular look. So the shape now is down here. So it's now down here. The molecule has turned, and then this is now where we are. So you can see it has been picked. Then the protein carrier moves with it, and then at the end of the day, we are going to see these molecules being released out here. So they have been actively transported across. At this particular moment, we can say that this thing is turning because of we had got the ATP being broken down to ADP, and then we have got the phosphate molecule here, molecule turning out with it, and then there is an enzyme here, ATPase. So that is the activity at this particular point. So we are having the energy, and then that at the end of the day, we are having the molecules being released, and then after that, this particular thing will turn again, so that it can go and pick more molecules. So this is how the molecules are actively transported across the membrane. So the last thing that we should remember out of this particular concept is, when we are addressing the exam situation, we have to remember that first, this active transport, it was happening against the concentration gradient, meaning that there are molecules that the cells need, and it needs them continuously, though they may be in the cell already, but we need them to be supplied continuously. Therefore, it will force the cell to get them outside its membrane, even if they are less outside, so they'll be ferried by the energy. So there is that movement against the concentration gradient. Very important for us to know when we are discussing the active transport. Then number 2, we should be remembering that in that process, therefore, there must be the use of energy so that we can facilitate this process. So we can be asked that what are the actually the evidence, or what are the requirements for the active transport to be taking place. So number one thing that we have to know, that for the active transport to take place, we know that number one, it must take place, it only take place inside living cells. Very important for us to know that active transport will only take place inside living cell. Why is it so? It is only living cell that can provide the energy in the form of adenosine triphosphate. So living cell can carry out respiration and store energy as adenosine triphosphate. That is one thing that is very important for us to note. Then number two, we have to know that this particular process will be facilitated much in those cells that have got a lot of energy. So it is going to be most, it's going to happen in many cells that have mitochondrion. Why is it so? Now, mitochondrial, we know that this is the cell powerhouse. This is where most of respiration take place. So if a cell can actually have got a lot of mitochondria, this will be facilitating active transport so much, because those cells will be having the correct amount of energy that is needed for the process to take place. So it's good for us to note that active transport is facilitated more in the cells that have got a lot of mitochondria. Apart from the cells being alive, there must be mitochondria. Then it is also good for us to note that if any case we have got anything that can facilitate respiration, that particular thing will be facilitating the active transport, like presence of oxygen, like presence of oxygen, and also high temperature and high temperature. These ones will be increasing the rate of active transport. So it is very much important for us to know the biological concept behind these two molecules being necessary, or two requirements being necessary for the active transport to be facilitated. Number one, oxygen is a requirement for respiration, and respiration normally releases for us the energy that we need for the active transport. So whenever oxygen is available, that is, whenever the cells are carrying out aerobic respiration, the active transport will be highly facilitated. Now, temperature, we know that apart from these particular molecules being taken up by the carrier here, they also have to move and reach the carrier. So for them to move, they normally basically store their own energy that make them move. But I want us to be very clear that the energy that they store is not what is going to make them to pass across. It is the energy that is coming from the cell. But for them to reach the site here, they must move. And they move easily when the temperature is high, because of the energy that they store. They can be facilitated. So they can quickly bind with the shape of the protein carrier, so that they can easily be ferried across. So therefore that one make the temperature also to be a facilitator for the active transport. Then this process can also be stopped. It can be inhibited. So this is also a factor to know that this one can also affect the rate of active transport. So we can talk about a metabolic poison. Metabolic poison can inhibit. Now, actually, inhibits is a term that we are familiar with. It's actually to slow down, to stop. So the metabolic poison can always stop this process. Like we have got the common metabolic poison in the cell, that is, we have got what's called cyanide. Cyanide, for example, cyanide. This is a common metabolic poison. It can stop active transport from taking place. Why? Now, if any case we have got this poison in the cell, this particular poison will stop the enzymes from working, especially ATPase. So when this ATPase is not working, it means that we cannot get the energy from adenosine triphosphate, and therefore that process will stop. So this is one thing that can actually stop the active transport from taking place in the cell. So that is one thing that is very much important for us to really note in this particular chapter or this particular lesson of today, and I want you to remember the hint that we have. Good afternoon and welcome to this session. Last time we talked about the active transport and the fission processes. Now today I want us to take this endocytosis and exocytosis and get to know what happens in this particular process as a means of transport. Now the fission and active transport, as we were talking about, it was the movement of the smaller molecules. Like we can talk about the smaller ions, or we can talk about the soluble molecules and the gaseous molecules. Those were smaller things. Now we realize that a lot is happening in the cell or outside the cell, and therefore we can also have the cell producing a slightly larger molecule, and then they want to release it out, or they want to take up molecules that are slightly larger as compared to the ones that we have been talking about. So how do these particular molecules move in? So as we were explaining the other time, endocytosis, this is when larger molecules are moving into the cells. And this one happens when we have got the cells developing. These molecules are forming something like a vesicle that can be coming together with the membrane of the cell, and then the cells take them up. So when the molecules are moving into the cells and they are slightly larger molecules, they are moving in by the process of endocytosis. A good example we can see when we are talking about the white blood cell. This is a very common thing. White blood cell, we talked about this earlier or lower level of learning. We said that white blood cells help us to protect our body against infection, and we saw that what happens, the process was referred to as phagocytosis. So what did we learn? If you look at the cell here, the white blood cell, like the phagocyte, we can have it developing a membrane. A membrane can have this shape. So if we have got bacteria, molecules of bacteria here that it wants to attack, you will realize that it will develop into a vesicle, like a shape, that will come into close contact with the bacteria here, and then this particular bacteria will move in. So you can see that this particular thing, at the end of the day, we can see that it has formed like a vessel inside here. The bacteria is moved in like that. So that is what has actually happened. So it's very keen for us to know that there is that particular vesicle that has been formed on the membrane, and then this thing does not just bind with the membrane, but it can pass in as a vessel on its own, and then this thing inside here, like we learned, will be eaten up by the enzyme. Like an example is lytic enzyme that will be eating it up. So you see, this thing moves into the cell. And how did it move? When they came here, the cell developed into a shape, like a phagocyte cell does. And then after that, there is a new vessel that is formed that will be carrying it into the cell, like we are seeing. So when it is happening like this one is now what we are calling the endocytosis. So this is one thing that I want us to note. Now this process, just like the active transport, it will happen when this cell has got energy. So the cell has got a mitochondrion that is going to provide it with an energy to carry this particular process across the membrane. That is very important for us to note. Now, we also know that for these cells here, we have got cells that can be releasing some larger molecule, like they talk about the steroid, the hormones. Like we have got the cell, we can talk about, if I've got a cell here, and then there is a release of particles here. And a good example we can talk about, like when you talk about the amoeba, for example, amoeba. These things can be staying in fresh water, and therefore they can be absorbing a lot of water. So there is a vesicle here that can be formed, the vacuole. So vacuole here can be taking this particular water, absorbing it up here. And since this cell here has got a weaker membrane, if this process continue, it can burst up. So what happens? This vacuole here is now acting as our vesicle here. It is a vessel. A vessel. So it will be moving. Once it is full with water, it has got the ability to move. So it can move. So it can say that it moves here and then binds with this membrane, and we can see it releasing water. So at this point, we will be talking about exocytosis taking place. So you see, we are taking up molecules from within the cell, and then we are moving with it to the membrane, and then we can now release them. This is now also how the enzymes are being produced, and also how the hormones are being produced and released out of the cell, so that they can go and work somewhere else. Like when you talk about, a good example is the hormones that is found, the glands like the pituitary gland, This one can release for us hormone ADH, antidiuretic hormone. So this hormone, we know where it works. So the moment this particular gland here has produced this particular hormone here, there is a vessel that comes and then release it into the blood. So it is released in the blood, whereby the blood will be transporting it up to the kidney where it's going to function. So you see, we need this particular two process here to help us in taking up molecules that are slightly larger, and also the molecules that are slightly, larger into the cell and also out of the cell. So that is very important for us to note here, that these particular processes normally take place, and then they need energy in terms of transporting of these particular materials either across the membrane or within membrane. So that is the last bit that I want us to note under this particular transport. So next time we meet, we'll be talking about gaseous exchange as